Okay, let's go through chapter 10. Okay. Sorry about the helicopter. I don't know what the hell's going on, but hopefully it doesn't show up in the recording. Okay, so sensory receptors. Okay, basically, let me try to explain this simply. One of my favorite movies is The Matrix, right? Where you're actually plugged into Matrix and the brain is just basically receiving electricity in different places, and that's what you perceive as the real world. That is actually true. Okay, so not that we're actually in a matrix, but that's how you receive information. Okay, so basically, sensory receptors transduce the different forms of energy in the real world into the electrical signaling of nerve impulses. So light is changed into nerve impulses. Sound is changed into nerve impulses. Okay, touch, balance, etc., etc., etc. Everything is uh, changed into electrical signaling of nerve impulses. Okay, the different modalities of sensation, sound, light, pressure arise from differences in neural pathways and synaptic connections. Okay, so if the optic nerve delivers an impulse, the brain interprets it as light, even though the impulse is the same as it is for hearing. Okay. All right, the functional categories. So the importance is the type of signal they transduce. Okay, chemoreceptors detect changes in the chemical environment. Photoreceptors, okay, sense light. Thermoreceptors respond to either cold or heat. Um, Thermoreceptors only respond to a certain range of cold and heat. Below that or above that, what you sense is actually pain receptors. Okay, mechanoreceptors are stimulated by changes in mechanics. So touch, pressure, uh, vibration, all of these are mechanoreceptors. Okay, all right. Those receptors are pain receptors. Okay, they respond to chemical changes in the environment that symbolize pain. Okay, um, the perception of pain can be enhanced by emotion, concepts, expectations. Okay, um, and how we consciously deal with pain. The pain reduction depends mainly on endogenous opioids. Okay. Okay. Uh, Receptors can also be classified by the type of information they deliver. Proprioceptors measure body position. So, like they're usually stretch receptors and to tell you exactly what position your body is in, even if the eyes closed, so you don't know where that you can't see. Okay, cutaneous receptors, pressure, touch, cold, heat, pain, special senses. Okay, okay. The last way we classify things, external receptors respond to outside, internal receptors respond to inside. Okay. All right. This one is kind of important. I mean, those stuff you probably covered in anatomy, so I kind of skipped over it. Tonic and phasic receptors um, is one of the concepts that we deal with in physiology. Okay, receptors can be characterized on how they respond to a stimulus. Okay, um, phasic receptors respond to a burst activity when a stimulus is first applied, but quickly adapt to the stimulus by decreasing response. Like touch receptors do that a lot. So for those of you guys who are still wearing a watch, you guys probably don't feel your watch or your ring and stuff. But if you initially put it on, you do, right? And then you slowly phase that out. The other example of phasing receptors is if I put your hand in hot water, okay, not like scalding burning water, but it's like warm water. You feel initially warm, and then you slowly phase that out, like swimming pools and stuff. Okay. Okay. That way, it allows our brain really to kind of go, okay, this is the way it is, and then just phase it out and not constantly have to deal with that message coming in. Okay, so that allows us to deal with changes to the environment and not what the environment is. So you want to know another funny version of this is the frogging boiling water concept, where frog or crabs or stuff, where you put them in water and then you turn on the water and they don't know they're boiling to death and stuff. It's basic receptors. Okay. All right, tonic receptors maintain a high firing rate as long as that stimulus is applied. Okay, and these are slow. They're called slow adapting receptors as well. So no matter when the stimulus is applied, you keep getting stimulus, like equal action potentials until the stimulus is withdrawn. The phasic receptors, you have this phasing out process. Okay. Information from a given nerve fiber can be only experienced as one stimulus type. Okay. So the sensation produced by adequate or normal stimulus is the one your brain will receive. So a punch to the eye is perceived as a flash of light because your brain is used to receiving light from your eye, and therefore the sensation produced is the one that your brain receives. Okay. Okay. Okay, receptors behave very similar to dendrites of neurons. They generate depolarizations. So again, you have to cross over this threshold, okay, to receive sensation. Like, so you cross over a threshold and it fires, okay. Okay. All right. Passilian corpuscles are phasic receptors. Okay, so if pressure is maintained, the generator potential is diminished. Passilian corpuscles are what allows you to feel like your watch and stuff like that. It's by Fitbit, and you slowly phase it out and stuff. Okay. Okay. In tonic receptors, the generating potential is proportional to the density of stimulus. Okay. Okay. So here, tonic receptors. Okay. The receptor potential, there's a threshold, and as long as you cross the threshold, you get the higher you cross the threshold, the more action potentials you get. So you can see that here. Okay. 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 Cutaneous receptors, pain, cold, heat, are naked dendrites of sensory neurons. Okay, so touch and pressure receptors have special structures around the dendrites. So you can see those here. Okay. Oops. Okay. Melthus corpuscle responds to changes in texture, the free nerve endings, touch, sorry, keep moving this, touch, pain, hot, cold, Ruffini's endings are stretch receptors. The Pacinian corpuscle, the thing that looks like onion, that is the deep pressure vibration receptors. Okay. Okay. There are many more receptors that respond to cold than to hot. They're located very close to the epidermis. Okay. They're stimulated by cold and inhibited by warm. So the temperature range of the response is 8 to 28 degrees Celsius. So colder than that, it's pain. Okay. So all right, warm receptors are located deeper in the dermis. Okay. Okay. And again, these receptors are limited. They're not pain receptors. Okay. Hot receptors is the pain receptor. This is also called a capsaicin receptor. It responds to capsaicin. Capsaicin is the product that you find in chili peppers. Okay. Mammals are not actually supposed to like capsaicin, which is why those plants actually invented capsaicin in the first place. Our enjoyment of hot sauce and stuff is actually a learned response. Okay. 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 Other pain receptors, those receptors can be myelinated or unmyelinated. Okay. So sharp pain, no persistent pain. Usually that's more external, that's more internal. Okay. It's sensational. Skip. Okay. A receptive field is an area of the skin that, when stimulated, changes the firing rate of a neuron. Different regions of your body have different receptor fields. Okay, so the way to explain receptor field is a 2-point threshold. Okay, so and you know, we do this in the lab where you poke each other with either one pin or two pins, and you see where on your body you can recognize one pin or two pins the most. Okay, the best place is your fingertips. Okay, their fingertips are very, very, very sensitive. Okay, because each neuron only controls one small tiny area of your fingertip. So to set off that, you can only work one neuron at a time. So that can set off two neurons, that can set off one neuron. If that circle was bigger, then it's very easy to poke in the same area using two pins. Okay? Let me just find what that means. Okay. If I put two pins in a blue circle, I get one touch. If I put two pins in the red circle, I get one touch. But it's very easy to put two pins in the blue circle at the same time. It's very hard to do two pins in the red circle. Okay, so this areas of the red circle are going to be much more sensitive to 2-point discrimination than areas with blue circles. Okay, so where you have red circles, the tips of your fingers, okay, your tongue, okay, and those are 2 main places, okay, where you have the blue, okay, the back of your legs, your lower back, okay, you touch things with those, but not very often, and not very often you need to distinguish between 1 touch and 2 touch. Okay, you don't need sensitivity in those areas. So those areas of the body are one neuron controls a large area. So like you have bigger fields. Okay, so the bigger fields, less neurons control that area. If you look up something called the sensory homunculus in the cerebral cortex, you'll see like the hands take up more area than like your whole entire leg, thigh area combined. Okay, and the reason why is we have the distinguished feeling and touch with our fingertips far more than our backs. Okay. Okay. So important is embracing the raised dots and Braille symbols. That's why, you know, if you're trained to read Braille, you can touch with your fingers, okay? But they can't like touch with the back of the legs or anything, okay? Okay, so first finger, thumb, this is the most sensitive area, okay? Palm of their hand, less, big toe. There's actually a lot of sensitivity in the big toe because, you know, you have to grip things a little bit, but back and calf and thigh, okay, very little, okay? All right. Okay, so receptors, so this concept of lateral inhibition, okay, let's go back to the circles. Okay, so clear all the drawing. Okay. So circle, circle, circle. Okay, if I put pressure on the center circle, I can actually set off not only this receptor, but this receptor and this receptor as well. But I'll put more pressure in the center. So not only will this center one register as touch, it will inhibit this one and that one. Okay? It will inhibit, I'm gonna draw a closed circle. It will inhibit that one and it will inhibit that one. And therefore I only feel it in this point here. I inhibit these two and I feel it there in the middle. And that inhibition is called lateral inhibition. It makes the borders sharp instead of fuzzy. Okay. Okay, so the receptor that's most strongly stimulated inhibit those around them. Okay. 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 Taste and smell. Taste and smell are chemoreceptors. Okay. They respond to chemicals that are dissolved in food and drink and other stuff. Okay, but the reality is they're the same receptors. In fact, um, from the molecular biology standpoint, they're the same. You have taste receptors turn into smell receptors, and smell receptors turn into taste receptors, just based on where they're expressed in the body. Okay. Um, okay. So taste. Okay. So here. Okay. You have taste receptors. Okay. Actually, they don't really look like this, but the taste pores. Okay. These are actually on the little entry to the side of the tongue. They have little taste pores, those little taste hairs, and these set off these nerve endings right there. Okay. Blah, blah. So, okay. Let me explain the different categories of taste. Salty, sour, sweet, umami. Umami is turned on by glutamate. It's the taste of meat, and bitterness. Okay, so there used to be this concept called a tongue map, and let me show you what that means. Okay, please note that I can't draw worth anything. Okay, so that is my crappy tongue. Okay, I'm sure some of you have to remember this in probably elementary school, middle school, but there's an area like sweet here, okay, uh, salty here, sour here, bitter in the back of the tongue here, okay. I'm sure if you Google that, like taste map, they'll put up, you'll find that picture. That's not a real thing. That was a misinterpretation of a really old German paper that the people who were reading it didn't really understand German really well. But somehow that got codified and solidified into science, even though it's not real. So hopefully that makes sense to you. Okay. The other thing that gets solidified into um, science, but it's not really true. The three primary colors are not red, green, blue. Okay, the three primary colors are cyan, magenta, and yellow. Okay. Okay. And if you don't believe me, check your printer cartridge. All right. Okay. So here's how salty works. Here's how sour works. Here's how sweet and umami works. Okay. Um, this is how bitter works. They should have a sixth category eventually for fats. They haven't, officially worked it out, so it's not on the categories, but they will in future editions of the textbook, hopefully. Okay. 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 So how taste works. What we generally need to understand is things or taste are things that we want. So we want to taste sweetness, okay, because that's how the sugar is coming in. And for the food, same thing for salty. We need salt. Okay. Sour is acidic acid, basically. Sour is the taste of acid, so anything acidic. Okay. Umami is the taste of meat, obviously. Bitter is actually a very important concept. Bitter, we are extremely sensitive to bitter tasting things. Um, Bitter is supposed to be toxic. It's supposed to be a sense of toxin. Like, again, like capsaicin, bitter is a acquired taste. It is like we drink coffee because we like the after effects of, the stimulation of coffee, not the bitter taste of it. Okay. 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 Here's your sense of smell. Okay. Again, you have, this is the nasal cavity. So you have mucus that covers the nasal cavity, and things dissolve in that mucus, and these receptors pick it up, and they go through the cribriform foramina here, and eventually into your olfactory bulb. Okay. Okay. Okay, odor receptors. Okay, there are 380 genes that code for 380 different olfactory receptors. One odor receptor stimulates one protein, and therefore we can have different combinations of olfactory receptors. Okay, a lot of human odor receptors don't actually work, but mice and stuff, they can detect like thousands of different smells and stuff. Okay. 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 The vestibular apparatus is located in the inner ear and consists of two things. There are two types of equilibrium. One is the linear acceleration. One is the rotational acceleration. So linear movement, okay, and spin. Okay. Okay. The vestibular apparatus is controlled by the vestibular nerve, is the branch of the vestibulocochlear nerve. Okay. So this is a vestibular nerve. This is the auditory nerve or the cochlear nerve. Usually it's called the cochlear nerve now. That's probably the older term for it. Okay. Uh, okay. So they're sensory nerve cells. Okay. 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 So how your sensory nerve cells work. Let me show you this picture. Okay. So here is your inner ear. Your inner ear, there are these little calcium stones called otoliths on this membrane here. This is affected by gravity. Okay. So I tilt my head forward. The rocks are affected by gravity pull forward, and what happens is they pull the hairs down with it, and that causes more action potentials. Okay, it depolarizes the membrane faster, so more action potentials. If I tilt my head backwards, it hyperpolarizes, and that means less action potentials, and that is how I tell which way I move my head. Okay. Okay. So the unicle is horizontal, so side to side. The sagittal is vertical. Okay. The semicircular canals. Okay. You can rotate one of three ways. You can rotate XY. You can rotate XZ and ZY. Okay. Okay. So the rotation. Okay. So if I'm still, the hairs, the copula inside my semicircular canal isn't moving. When I move initially, the movement of the endolith is actually backwards initially, okay? Because my head moves first and then my endolith moves. So like that movement will bend the copula the wrong way, and eventually it will catch up, okay, etc., etc. And like, I think I explained this. I think it's explained in anatomy, so I don't explain this really in detail, but if you need help, I'll be happy to explain it again. Okay. Okay. When you suddenly stop, the fluid is still moving in the opposite direction. Okay, and that the eye movements uh, are moving toward the opposite direction of the spin to maintain a fixation point. Okay, when the body comes to stop, the copula is still moving and the eye movements are still moving. Okay. Okay, and then here, that. Okay. Okay, sound waves. Sound waves are characterized by frequency, which is measured in hertz. Okay, 1 hertz is by definition 1 cycle per second. The higher frequencies have higher pitches. Okay, their pitches and frequencies are almost the same. They're not exactly the same, but they're pretty close. Okay, so we have sound wave. We have two things. Okay, one is pitch. The other is volume or loudness. Okay. Um, okay. So frequency is which sound wave we're hearing. The loudness is how loud the sound wave is, the amplitude of the wave. Okay. The human optimal range is 80 to 0 decibels. Okay. Above that, you get hearing loss. Okay. Okay, so sound waves go through the external ear, through the eardrum, through the ossicles, the malleus, incus, and stapes. Okay, hammer, angle, stirrup. Okay, the vibration, the stapes is attached to the oval window, which transfers the vibration to the cochlea. Okay, the subpenius muscle stabs the stapes, and the intensity is too loud, so that little muscle here. Okay. Subpenius is, there's another little muscle, subpenius right there. Okay, and this one as well, the tensor tympani. Okay, it prevents those bones from damaging. Okay. Okay, so there you can see the stapes attached to the oval window. There's another window here called the round window. That's where sound comes out, not goes in. Okay. Okay, so let me explain how sound works. Basically, as sound comes in, you here, okay, you have a bunch of little hairs. So what they basically did with this image was actually unroll the cochlea. Okay, and so basically you have little vibrations, okay, that these hairs can pick up. Okay, blue. Okay. Okay, so when sound waves, okay, enter the scalar media, they vibrate, okay, and bends the oscillate. It doesn't bend all the cells, so it only bends particular ones that respond to the frequency. Okay. Um, okay. So okay. So the place area of pitch. The hair cells located to where the vibrations are displaced in the scalar media are stimulated more often. So when the vibration comes through, only certain hairs will vibrate. Those hairs that vibrate tell you what pitch you're hearing. Okay. Um, okay. And so you have longer hairs here, shorter hairs here. So higher and higher pitch. Okay. And as you get older, you lose these guys here, and therefore you lose the ability to hear high pitch. Okay. 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 So what else? How much that hair vibrates? Okay. The amplitude in which it vibrates tells you how loud it is. Okay. So slight movement, very low sound. Very large movement of that hair, very high sound, like loud sound, not high. Okay. So I don't want to confuse pitch. All right. So hearing impairment. There are two ways to screw up hearing. One is conduction deafness, and one is sensory neural deafness. Conduction deafness is blocking the pathway of sound somehow. Okay, you can fix that relatively easily. Okay, due to buildup of earwax, too much fluid in the middle ear, damage to the ossicles, damage to the eardrum. Okay, ossification of the inner ear so that they don't vibrate quite as much. Okay, this can be helped by hearing aids or cleaning out your ears and all this other stuff. Okay, so imagine if you stuck a cotton ball in your ear. That is conduction deafness. Okay, sensory neural deafness. Okay, the nerve impulses are not conducted from the cochlear to the auditory cortex. This can be damage to the hair cells. Okay, this may only impair hearing of particular sound frequencies and not others. So if I damage, like, since I'm not blocking sound, I'm stopping the vibration somehow, I'm stopping the nerve somehow, I can impair only, sometimes I can only impair certain sounds. Like I said, as you get older, you stop being able to hear the high frequency sounds, okay? So these unique cochlear implants and stuff, okay? Okay. Okay. So let's go back. Okay. Okay. Vision comes from light energy transduced into nerve impulses. Okay, we only see a tiny little portion of the EM spectrum. Okay, so you can see that here. Okay, so structures of the eye, okay, are located here. The internal anatomy of the eyeball is located here. Okay. So basically, the general pathway of the light through the eye, okay, you start off with the cornea. So the purpose, okay, I know I'm gonna say something that sounds like 1 + 1 = 20, but the light, the eye is not what you see with. Your brain is what you see with. The eye transmits light and reflects it onto the retina. Okay? So the pathway of light goes through the cornea, it goes through fluids here called the aqueous humor, it hits the lens, okay, the cornea and the lens bend the light, so it hits the back of the retina. Okay, and there's fluid here called the aqueous humor, and there's another fluid here called the vitreous humor. Okay, and it hits the back of the retina. Okay. Okay, the iris and pupil basically, the pupil is a hole to allow light through. We can dilate, sorry, we can constrict or dilate the pupil. Okay. 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 So, so, okay. So you can see the lens there. Okay. Uh, okay. The aqueous humor is fills the anterior and posterior chambers, okay, of the eye. So the front of the eye is a clear watery fluid that's secreted by ciliary bodies to provide nourishment to the lens and cornea. Remember, the lens and cornea are avascular, so they need nourishment, and they get nourishment from the aqueous humor. It it drains back into the sclerovenous sinus, also known as the canal of Schlemm. There's an H there that's in that they didn't put they spelled wrong. And inadequate drainage leads to pressure in the eyeball, and that leads to glaucoma. Okay. Okay. So if you build up too much pressure on the front of the eye, it puts pressure on the nerve in the back, and that is damage to the optic nerve. Okay. 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 So when light passes through one medium to the other, at different intensity, it bends. Okay. So the air cornea surface. Um, okay. Changing the curvature of the lens allows the fine control of the surface. The humors also contribute to the refraction. Okay. When you see the image is actually flipped upside down and flipped left to right in this process. Your brain actually flips it back. Okay. So we take the image, okay, the image is an arrow, right? The arrow is actually flipped upside down and actually reversed left to right as well. Okay. Visual fields. Let me explain the visual fields. Okay. So here's the field of view. Okay. Everything on the right side, okay, is sent to the left side of the brain for processing. Everything on the left side is sent to the right brain for processing. And therefore, the image. Unknown caller. Um, all right. Um, okay. Lens and combination. So our lens, our eyes are not designed to read books. Okay, they are designed to see things from far away. Like the focal point of our eyes is actually about 20 feet away from us. Um, we're designed to see things that are far away, and so as things come closer, the lens has to squeeze down to focus down on an object. Okay, it causes the lens to thicken and round up. Okay, I'm sure most of you guys are too young to, need reading glasses and stuff like that. But as you get older, the lens stiffens and it becomes harder and harder to accommodate, and therefore, that's what reading glasses are for. Okay. Okay. So there we go. Okay. So this is for distance vision. This is the relaxed version of the lens. This is for the reading, okay, or close-up vision. Okay, The visual acuity, blah, measures. So let me explain this. Okay. So this is normal. Okay. This is nearsightedness. Okay. This is farsightedness. This is normally the problem. So the lens corrects it. Okay. Oh, sorry. The nearsightedness is usually the problem, and so the lens actually fixes the problem. Okay. The astigmatism is the rays don't focus on a point. That's what the astigmatism is. Okay. Okay. The retina. Okay. So here's the retina. Okay. So we look at the retina. There is an optic disc. The optic disc is where all the blood vessels and stuff run through. Okay. And the fovea centralis, there's no blood vessels at all there. Okay. So that's visual acuity, complete lack of visual information. Okay. There are two types of photoreceptors, rods and cones. Okay. So rods look like rods, cones look like little cones. Okay. So light has to pass through all this, hit this membrane, and then the signal gets transduced here and sent back up this way. And therefore, there has to be a place where, let me just draw this in. Okay, so there has to be a place where the nerve runs back out the eye, and that is the optic disc. Okay, this right there is the optic disc because the nerve has to literally run out there and get in the way of the photoreceptors. So there's no photoreceptors in that area. Okay. Okay. Drawing. Okay. So rods, cones. Okay. Okay. Rods. Okay. Allow for black and white vision in low light. So if you like really, really dark room, okay, you see things in black and white. The other cool trick is rods are generally at the side, so your peripheral vision actually works in black and white, and your peripheral vision actually works faster than color vision. Okay. 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 Absorption causes rhodopsin to dissociate into retinal and opsins. This is derived from vitamin A. Okay. It's called a bleaching reaction. So this is so the story about carrots being good for your eyes and vision. That's the reason. Okay. Okay. So you cycle through retinol. Okay. So when light hits it, it then turns out trans, and then you switch it back, and then you use it again, etc. Okay. 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 Dark adaptation. When a person enters a dark room after being in the light, there are fewer photopigments. In the rods and cones, after about 20 more minutes, more visual pigments are produced okay, and therefore you can fire them easier, and therefore you can see better in the dark. So if you're literally in a light room and then it goes dark, it turns black all of a sudden, and then if you stay there and focus, you're after about a little while you start seeing more. You start seeing more and more stuff because you become more and more sensitive to the limited amount of light. Okay. All right. The dark currents. Okay. It's really weird. I always like when you think about how light works, you always think it works backwards than it actually does. So let me explain how this actually works. Okay. So here's in the dark. Okay. Um, in the dark, the sodium channel is always open, and therefore this is inactive. Okay. So what light does actually is you trigger all this, and ultimately you close the channel, and you close the channel, and that causes hyperpolarization, and that hyperpolarization is what you perceive as light. Okay, which is kind of backwards. You would think light sets something off. No, it actually doesn't. Light actually prevents something from being set off. But any type of on-off switch we can perceive as switch. Okay. So light actually stops the dark current, okay, in the photoreceptor. Okay. Uh, oh, okay. Okay. So once that goes off, okay, it goes through the bipolar cells, then the ganglion cells, and then to your through your optic nerve. The optic nerve is the axons of the ganglion cells. Okay. Um, Cones are less sensitive to light, but they allow for color vision and greater visual acuity. They're sharper. Okay. Um, the reason why they're sharper is because one cone always attaches to one bipolar cell, which attaches to one ganglion cell. Rods don't do that. Rods actually, multiple rods attached to the same bipolar cell. So it's easier to trip, but it's less accurate. Okay, so most humans have tetrachromatic vision. So we have three types of cones. Okay, short wavelength, blue, long wavelength, green, red, and medium wavelength, green. Okay. Okay. So we see in three different lights, right? RGB. Okay. The three colors are not RGB. The three different lights are RGB. Okay. Um, so, okay. Color blindness. Okay. Color blindness is missing one or more of the cones. And it's more common in men than they are in women, as the genes are located on X chromosome. Okay, so I'm sure you guys remember from earlier, men have one X. Okay, females have two. So men, if they lose one, they're screwed. Females, if they lose one, they have a backup copy. Okay. Most commonly, this involves either the red, green, color blindness. Okay, so they have trouble distinguishing between red and green, and see colors like purple and brown as different than normal. Okay, so let me show you what red and green color blindness is. Green. Okay, so if we can't read any of these numbers, okay, um, okay, if the first one doesn't see, you don't see the big letter A, then you're colorblind. Okay, just kidding, it's not A, okay, so don't freak out. Okay, but you should be able to see the colors, the red versus green. If you're actually colorblind, you can't make out the difference because you're missing one of the two cones. Okay. Um, so there's a wavelength. If you're missing one cone, like the red cone, you perceive what you perceive from the red cone with the green cone. So you will be colorblind. Okay. Um, one more cool thing before I end this. Okay. So most humans are tetrachromatic. There are females or trichromatic. There are females that are tetrachromatic. Okay. In addition to having these three cones, they have a fourth cone that is orange, and it's like literally right in between the green and red. So you can either have the red cone or you can have orange cone on the chromosome. So males only have one, so we perceive that as red. Females can have both because they have two X chromosomes, and those people are tetrachromatic, which means they can see into them four colors of light, and that means they can see much more shading of light of color than others. Okay. Okay. Vision is best at one point in the retina called the fovea centralis with the macula lutea. Okay, the fovea centralis is all cones. The macula lutea is mostly cones. Okay. Okay, so each cone has a one-to-one relationship with a ganglion cell. Okay. Okay, so only works in good light. Okay, so there you go. Virgins. Okay, so like I said, rods converge, so it takes signal from any of these three as light, so they're more sensitive. Okay, cones are less sensitive, but they're more accurate. Okay. Okay. The okay. Uh, Pupillary reflex. I'm sure you guys can read that on your own. Okay. What else? Okay. Last thing I want to cover is this. Um, the neural pathway for vision. Okay, we have something called binocular vision. So the right eye sees a lot of things that the left eye also sees, and so we have overlapping fields of view, and that is a binocular field. Okay, so that goes to both sides of the brain, and because they come from both eyes, we have a much better perception of where it is in the real world, and therefore we also use things like we know the size of the object. So the further away the object is, if I know the size, I can guesstimate where it is. But if we don't know the size of the object, it actually requires both our eyes to focus in on the object so we can get binocular vision and see the object and grab it. Okay? Okay. All right. So hopefully this makes sense to you guys. I'll stop sharing right here. And